Hi friends, good afternoon. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mohanty. Today we shall discuss about spleen. The spleen is the largest lymphoid organ in our body. It is a secondary lymphoid organ. Its network of trabeculae, blood vessels and lymphoid tissue provide an environment in which lymphocytes proliferate while old damaged erythrocytes are recycled. In today's video, the following points will be covered: location, surfaces, splenic ligaments, histology, blood vessels, lymphatic drainage, innervation and function. So first point is location. The spleen is found in the left hypochondriac region of the abdomen. More precisely, it is located posterior to the stomach and anterior to the left hemidiaphragm at the level of ribs 9 and 10. Medial to the spleen is the left kidney, whereas superior is the diaphragm. Inferiorly, the spleen rests directly on the left colic flexor. The spleen is a purple, fist-sized organ. This organ is covered by a fibroelastic capsule which allows the spleen to increase its size significantly whenever necessary. The spleen is an intraperitoneal organ. That means all of its surface except the hilum are covered by visceral peritoneum. Hilum is the location on the surface of the spleen. It is the point of insertion for splenic artery and splenic vein. The organs near to the spleen leave their impression on its surface, which together with spleen borders can be easily observed and described. This is about location of the spleen. Next is surfaces. There are various surfaces. One is diaphragmatic surface. Diaphragmatic surface lean onto the adjacent part of the diaphragm. This is the diagram of a spleen which look like a fist. And the spleen is having various surfaces. This surface is known as diaphragmatic surface, and the diaphragmatic surface lean onto the adjacent part of the diaphragm. It also bear impression from the ribs 9 to 11. The medial surface of the spleen has three areas of impression. The gastric area is the impression of the stomach, renal area is the impression of the left kidney, whereas colic area is the impression of the left colic flexor. Structure of the spleen. The spleen is the largest lymphoid organ in our body. It is an intraperitoneal organ. That means all of its surface except the hilum is covered by a layer of visceral peritoneum. Underneath the peritoneum there is a capsule that covers the spleen. The capsule consists of fibroelastic connective tissue. This diagram explains the internal structure of the spleen, and this is the capsule. The capsule covers the spleen and it consists of dense irregular fibroelastic connective tissue. This connective tissue contains contractile cells known as myofibroblasts. The contractile activity of these cells help the spleen to discharge stored blood into the circulation whenever necessary. In addition to this, the capsule also allows the spleen to increase its size whenever necessary. The capsule gives rise to a number of septa these septa are known as trabeculae. These trabeculae penetrate into the parenchyma of the spleen and partly divide the tissue. Next is, like other organ, spleen also consists of stroma and parenchyma. The stroma consists of a network of reticular connective tissue that provides support for blood cells and also for cells of the immune system like lymphocytes, macrophages and dendritic cell. Next is parenchyma. The parenchyma of the spleen is divided into two morphologically and functionally distinct compartments known as red pulp and white pulp. Red pulp and white pulp. The red pulp and white pulp, they are the two compartments of the parenchyma. They are separated by marginal zone. The red pulp and white pulp are separated by a marginal zone. This is the marginal zone which separate the red pulp and the white pulp. First of all, we will discuss about red pulp. The red pulp, it consists of splenic cord and vascular sinusoids. The splenic cord is also known as cord of Billroth. The splenic cords are cellular aggregations that are supported by reticular connective tissue, and the splenic cord contain different types of cells like blood cells, macrophages and plasmocytes. Other is vascular sinusoids. The vascular sinusoids are blood-filled space, and the splenic cord appear as stripes. The blood flow slowly through the vascular sinusoids. These are the vascular sinusoids. The blood flows slowly through the vascular sinusoids where they become exposed to macrophages from the splenic cords. So the red pulp acts as filter for removing the toxin from the blood before it enters into the systemic circulation. White pulp. The white pulp of spleen consists of lymphoid follicle, marginal zone and periarterial lymphoid sheath. The periarterial lymphoid sheath consists of central artery which is surrounded by a sheath of lymphoid tissue. The lymphoid tissue is distinguished into two layers. The inner layer contains only T lymphocytes, whereas the outer layer contains both B lymphocyte and T lymphocytes. The branches of the central artery, these are the branches of the central artery, are surrounded by lymphoid follicles. These are the lymphoid follicles. Two types of lymphoid follicles are seen in the white pulp. They are primary lymphoid follicle and secondary lymphoid follicle. The primary lymphoid follicle contain mainly small immature lymphocytes. When the primary lymphoid follicle mature and increase in size, it is known as secondary lymphoid follicle. The secondary lymphoid follicle differ from primary follicle in having a germinal center. The germinal center is the site where the B lymphocytes acquire the ability to produce antibodies. These are the three components of the white pulp. One is lymphoid follicle, another is marginal zone, next is periarterial lymphoid sheath. Follicle means lymphoid follicles. Two types of lymphoid follicles are seen, primary lymphoid follicle and secondary lymphoid follicle. Then there is a marginal zone. This marginal zone is between red pulp and white pulp. and next is periarterial lymphoid sheath, which consists of a central artery that is surrounded by a sheath of lymphoid tissue. So this is known as periarterial lymphoid sheath. Blood from the splenic artery enter into the spleen through the hilum. From there, the splenic artery divide and form many branches which enter into parenchyma following the course of trabeculae. These artery branches throughout the parenchyma and gradually decrease in diameter. In parenchyma, these arteries are surrounded by periarterial lymphoid sheath, and the artery which is present in the central region is known as central artery. The central artery divide and form many branches which enter into the marginal zone. From there, the arteries further move into the red pulp region where they transform into arterial capillaries. From arterial capillaries, blood flow into the splenic cord. In the splenic cord, old and damaged erythrocytes are destroyed by the macrophages, and from splenic cord, blood flow into the vascular sinusoids. Due to the lack of junction between the endothelial cells, only the normal and healthy erythrocytes become able to enter into the vascular sinusoids, whereas malformed and old damaged erythrocytes as well as antigens like microorganisms and cellular debris are destroyed by the macrophages which are present in the splenic cord. From vascular sinusoids, blood flow into the trabecular vein, and from trabecular vein, blood ultimately exits the spleen through the splenic vein. Blood flow freely in the vascular sinusoids. This type of circulation is known as open circulation, and open circulation is the characteristic of the human spleen. This is about the structure of spleen. Function of spleen. The most important function of spleen is to filter blood. The spleen is considered as the graveyard for all damaged erythrocytes because it removes them from the circulation. The macrophages that are present in the spleen destroy old and damaged erythrocytes, thereby helping in the recycling of iron that is present in the hemoglobin. Other important functions are: one is during fetal development, spleen is the site of hematopoiesis. Spleen is the site of hematopoiesis in fetus before the development of bone marrow. The spleen is one of the major sites for initiating and modulating immune response. Now we shall discuss about the splenic blood vessels. The splenic blood vessels include splenic artery and splenic vein. The splenic artery provides arterial blood to the spleen. During its journey, the splenic artery gives rise to a number of branches that supply a large part of the pancreas and stomach. The branches of the splenic artery that supply pancreas are dorsal pancreatic artery and greater pancreatic artery, and the gastric branches are short gastric artery, left gastroepiploic artery, and posterior gastric artery. These gastric branches supply arterial blood to a large part of the stomach. Next is splenic vein. Venous drainage of the spleen is occurred by splenic vein. The splenic vein also receives blood from inferior mesenteric vein. Then the splenic vein It unites with the superior mesenteric vein posterior to the neck of the pancreas to form hepatic portal vein. Next is lymphatic drainage. The splenic lymph node lie at the hilum. It receives lymph via perivascular and subcapsular lymphatic vessels. From there, the lymph is drained to the superior pancreatic lymph node, and from pancreatic lymph node, the lymph is drained to the celiac lymph node. Next, innervation. The spleen is innervated by autonomic nerves from the celiac plexus. The celiac plexus provides the spleen with both sympathetic and parasympathetic nerves, and these sympathetic and parasympathetic nerves, they constitute splenic plexus, which reaches the hilum and travel along the splenic artery and its branches. Hello friends, this is about spleen. Please subscribe this video, and also please don't forget to subscribe my YouTube channel Sam Online Class.